Right, this is the third video in our Unit 3, which is the third 9 weeks material. We're going to finish protists at the beginning of this, and then we're going to run through fungi. So that was our third 9 weeks was bacteria, protists, and fungi. We left off with dinoflagellates. Dino means a two, flagella is obviously a flagella. So you can see here we've got two flagella. They're part of the alveolate, which is part of the SAR supergroup. These guys can be heterotrophs, phototrophs, or mixotrophs. And we spent quite a bit of time talking about the fact that these guys cause red tides and that red tides can be very detrimental to both health and economy. So Florida beaches have been shut down fairly commonly due to red tide. We spent quite a bit of time talking about Plasmodium, which is an apicomplexan. It's parasitic and it causes malaria. So we went through this life cycle, and again, we have alternation of generations. So we started here with the mosquito bite, and the mosquito bites the human, and it releases sporozytes. So these are sporozytes. These are depicting them here and then over here as well, and they go straight to the liver. Once they're in the liver, they change form and they get released as merozytes. So these are merozytes that enter the red blood cells. And once they're in the red blood cells, they cause, this is where you get your symptoms. They cause fever and chills. They incubate inside the red blood cells, and then they leave as gametophytes. And we are in almost this whole cycle, we are in the haploid form. So from the merozytes, the gametocytes form, and this is where the mosquito bites the human and picks them up. So a mosquito is a vector, and this vector is a carrier. So part of the life cycles in humans, part of it is in mosquitoes. And once here, those gametocytes form into gametes, we get fertilization, and now we are diploid. So here we go into the diploid form quickly. So from fertilization, we go into diploid, and then we undergo meiosis, and after meiosis, we form these sporozytes. Actually, this is an oocyst. So the zygote, we go to an oocyst, and it forms the sporozytes. We said malaria, 450,000 deaths each year, and we talked about sickle cell and how sickle cell is a mutation that can help you survive malaria, but it also has its own disease pathology. So it's got pros and cons depending on where you live. From here, we talked about Paramecium's ciliate. So this is part of the ciliate group. And we very briefly, we looked at the anatomy. And again, I'm going to look at overviews here because we are reviewing for the purpose of the final. So we talked a lot about the fact that these guys can do conjugation and then they undergo asexual reproduction. We didn't necessarily look at or require you to know this life cycle in detail, but it's good to understand that they can use conjugation to get their swap their genetic material. So this is where we're going to get your variation. That they have asexual reproduction. Then they also have two nuclei. So we've got a large nucleus and a small nucleus. That's a good thing to know. And that they use cilia to move, hence the name ciliates. This was the second half of the cladogram. So we talked about now amoebas. One of the things we didn't mention was that forams and radiolarans actually have pseudopods, which technically makes them amoebas. But we talked about most of our amoebic life here. Here we had green algae, red algae. We also talked about slime molds. We also talked about the entamoebas, but I'm going to skip that because, again, that was kind of some of our details. Red algae, these guys have a red pigment that can absorb blue light so they can grow deeper. They are multicellular. It's one of the few examples that we have that are multicellular. Green algae, again, we have precursors to plants here, and some argue that algae should be in the plant kingdom. In Uniconta, we have the, whoops, shoot, come back. Tubulinids. And this is where we have amoebas. This is what we think of when we think of amoeba. We think of that pseudopod as oozing forward and then the rest of the organism pulling in behind it. They're predatory, they're heterotrophs, they are terrestrial and aquatic, and we get these tube-shaped pseudopods. This one was one you needed to know quite a bit about for the practical. All right, that takes us to fungi. And we said in fungi, we have 5 groups. We have chytrids, we have glomeromycetes, we have zygomycete, we have ascomycete, and we have basidiomycete. All fungus are heterotrophs that absorb their nutrients. And we said they release digestive enzymes into the environment around them that breaks down whatever's around them, and then they can absorb it. Now, we said most fungi, not all, they have chitin in their cell walls. And they have a filamentous body plan in terms of hyphae and mycelium. And we'll look at that on the next page, I believe. So here I have an example. The mycelium are the filaments that exist below ground. And this is a fruiting body. We call this a fruiting body. This is the structure that's above ground that produces spores. Now, an individual fiber is called a hyphae. If you put all of them together, it's a mycelium. We said that we can have 2 forms of overall body plan. We have septate, where the nuclei are separated, but we have these little septums with holes in the middle. You can see the holes. Those allow organelles to move through. Or we can have cenocytic, where there are no septums. We then started to look at the overall body plan. All of the fungi go through this overall body plan where we have a fusion of the cytoplasm. We have 2 different hyphae come together, and plasmogamy, they fuse, and we get this heterokaryotic stage where you have a structure that has 2 nuclei. It undergoes meiosis, it germinates, and then we reproduce. But so we have this new heterokaryotic stage, and we're going to look at each of these in detail, and you'll be able to see what we're talking about. You have the breakdown here. So we have chytrids and fungi, and animals, they went in opposite directions. So the fact that they are both multicellular and some of their characteristics are due to convergent evolution. Here you have again the 5 groups, and it tells you how many. You can see that ascomycetes are by far the biggest, Basidio are the next largest. There aren't a lot of glomeromycetes, and chytrids and zygomycetes are about the same. Chytrids, the thing to know for the final is that they have flagellated zoospores. And we talked a lot about what happens with chytrids and the amphibians, especially frogs. Our first, oh, almost forgot about these guys. So these are my glomeromycetes, and they have what are called arbuscles, where part of the fungus invaginates into the plant cell. So they are symbiotic mutualists. That's what glomeromycetes are known for. So the fungus help the plant get their nutrients and minerals, and the plant provides the fungus with glucose. So there is that mutualistic relationship. All right, here we go. We've got our zygomycetes. So here's my plasmogamy, where you get 2 hyphae that they have a pheromone that allows them to detect if they're compatible, and if they are, they fuse. And what we get then is this heterokaryotic condition right here in the middle. And you see a picture of it down here. This is called a zygosporangium. This is very characteristic for zygomycetes. This thing is super durable. It can withstand extreme conditions. And that at some point we have the karyogamy where we get fusion of those nuclei, and then we undergo meiosis and we continue with our life cycle. But again, this heterokaryotic stage is really our unique portion. And this is similar to an endospore. We said the bacterial endospore. Here's the ascomycete. Ascomycete translate to cup fungus. And that is named because of the ascocarp, and it looks like a cup. So again, we have our two hyphae. We get plasmogamy. Here, my dikaryotic or my heterokaryotic cells look a little bit different. But again, we have karyogamy where we get our fusion. Meiosis. Now, one of the things we did, especially for practical, was we did a lot of sketches of these, and we looked at these asci, which are the long finger-like projections, and the ascospores, which are the individual little dots you see there, and then these get dispersed. So one of the things for the final that you really want to focus on, this is what makes all of the fungus have the plasmogamy, karyogamy, meiosis. But this is what makes the ascomycetes unique, is that we have the ascocarp, and we did a lot of sketches of asci inside of ascocarps with the spores. Yeast are also ascomycete. They are one of our exceptions. They reproduce by budding, and this was an example of how the yeast reproduce. Then we went to basidiomycetes again. Oh, darn it. Why I keep hitting that? Plasmogamy. Son of a gun. Karyogamy, meiosis. And the unique part here for basidiomycetes, and by the way, these are the club fungus, and we looked at the fact that these guys have gills. So we did a lot of sketches of gills under the dissecting microscopes. They formed these basidium, and apparently early naturalists thought these looked like clubs, so that's where they get their name. But these are basidium, and they produce basidiospores. So here is an up-close electron microscope picture. Here are those gills lined with basidia. That's also unique. So it's important to know for the three fungi groups of zygomycete, ascomycete, and basidiomycete, what makes them unique. So here was a breakdown, again, flagellated zoospores for chytrids, and it's actually chytridiomycota, but we just say chytrid. Zygomycetes, you get that zygosporangium, mutualists, so know that these are mutualists. We have the ascocarp and we have the basidiocarp. Ecological roles, we looked at how fungi operate within habitats, and we said that they are decomposers often. That's a very important role. And that part of the reason scientists believe we formed as many fossil fuels as we did at that last great extinction event is because there was a lack of fungi to break them down. We looked at examples of parasitism, ergot rye with the Salem witch trials, chestnut blight. And then we looked at mutualist. So we said we could be ants and their fungus farms. This was endophytes. These are so in roots we have arbuscles, and in leaves we have these endophytes. It's a mutualistic fungus, and we looked at how that increases the resistance of the leaf because it has that mutualistic. Last thing, and this won't be on the final, but we looked at lichens, and we said those are algae and fungi together. Okay, that gets you through Unit 3, and Unit 4, we have plants and animals, and that'll be the end of our review videos. All right, thanks for watching. See you in the next one.